So definitely transports gases. That's real important. Also, you know, what you eat is not going to get to your tissues without going through the blood. Also, metabolic waste, kidneys, filter out the metabolic waste which will circulate in the blood. Enzymes, hormones, yeah. Also, the pH of the blood is very critical, and it contains all the stabilizing electrolytes and things like that. Clotting mechanism, when you cut yourself, you quit bleeding unless you cut an artery. Also, because of the white blood cells, defense against pathogens. And also, something you can relate to when it gets hot, especially if I go back east, helps stabilize our body temperature by sweating, by bringing the heat to the surface and allowing it to basically evaporate. So, I don't know, there's lots and lots of things that you can say about functions of the blood. Doing all these things, without it, you know, obviously you don't survive. So blood is extremely important. Remember, it's a type of fluid connective tissue. A word about blood, and I'm sure you know about this stuff anyway, if you have any sort of vague, even medical background at all or anything. Whole blood consists basically of two parts, the formed elements and the liquid part, though. So if you take blood and spin it in a centrifuge, spin it around real quick, it'll separate out into two components. But the formed elements of the blood are the red blood cells, which are by far the most numerous. The white blood cells, and we'll get to those tonight, these are the infection-fighting cells to a certain extent, and also these little fragments of cells called platelets, and those are involved in clotting. So the formed elements, almost half, this is 37%, awfully low. Roughly around half of whole blood consists of platelets, white blood cells, and 99.9% of the red blood cells. The red blood cells, of course, carry oxygen, so, you know, there's a huge amount of need for red blood cells. However, if you separate it out into, if you take blood and you spin it, it separates out into two layers. In the top layer, which is kind of clear, the color, sort of slightly yellow in color, that's the plasma. The bottom part are the RBCs, are the formed elements. So the plasma is mostly water. It's got some protein in there, and, you know, other solutes, but it's mostly water. So if you take blood and you spin it in a centrifuge, you basically get something that looks like this. You get the plasma at the top. The RBCs, the red blood cells, pellet out. They go to the bottom because they're heavy. And then if you look real carefully at a sample of blood, right along this interface between the plasma and the RBCs, there's a little area, hematologists, lab people who work with this stuff, call this area right here the Buffy coat. And this is where you find the white blood cells. I used to work in an immunology lab, so I good at this stuff. So anyway, so it's mostly water. It should be clear, yellowish, maybe the color of urine. But if it's cloudy or something like that, that could be a problem. One of my jobs that I've had when I was working in a hospital was I worked in a lipid lab. And we dealt with patients that had lipid disorders, and we would get fasting bloods from our patients, and the plasma would be like the color of milk. So these people had extremely serious lipid clearance disorders, and many of them died at a relatively young age just because their arteries clogged up. So for most of us, of course, the plasma is going to be clear. Well, you can see what it does. We're not going to go into all the details and all the gory details of what you need up here. But it's mostly water. There's, you know, all kinds of ions and, you know, other things in there. You don't need to memorize this list, certainly. You just need to know that plasma is the liquid part of the blood, and it's mostly water. The proteins that are there, albumins, globulins, you know, the clotting proteins and things like that. And I know that I'm sure that at some point in time in your life, you had blood work done. Now, you know, they take a little, you know, reading cubital veins when you have to learn. Usually, you know, stick a needle in there, and they draw out various colored tubes. The colors of the tubes, they got stuff in them that, you know, maybe the blood they want it to clot, maybe they don't. You know, there's all kinds of stuff in there. So anyway, so that's basically the breakdown between plasma and formed elements. So, liquid matrix is plasma, formed elements are the blood cells. Relative amounts here, males, maybe 5 to 6 liters of blood. Of course, that depends on the size of the individual. Females, 4 to 5 liters. But this is critical, the pH. The pH of blood cannot vary more than just like 1/10 of a unit. If it goes down or up, that's a medical emergency, acidosis, alkalosis. And I'm sure, looking around this room, and no, I'm really teasing about this, of course, but there are several people in this room whom I'm sure have never given blood because you're just too small, right? The rules are you have to, was it 115? Is that what it is?
Yep.
I think, is that what it is, 115 pounds? Yeah. And the reason for that is they figure if you're way less than that, this volume is going to be low. And if we take out a unit, it's kind of draining the tank more than we'd like to. So, but if you're bigger than 115, yeah, you get blood. Right? And we're not going to talk about blood groups, so I'll mention it. We're not going to talk about it. Yeah, it's a composition of blood. So I kept going on this. Don't worry, it's the same slide. This is just, again, the breakdown. Many of these plasma proteins are involved in regulation of osmotic pressure. Some of these are involved with antibodies and stuff like that. Here's the ones that are involved with the clotting reaction. But anyway, I would just generally for the test, what do I need to know? Just need to know the basic breakdown of blood, what the elements are and what the formed elements are, what the liquid parts are and things like that. You should be fine. This chapter goes into more detail than we need, but just generally speaking, this is fine. These are the formed elements: RBCs, WBCs, and platelets. We'll talk just briefly about them here for a minute. The red blood cells, of course, carry oxygen. The white blood cells are involved in infection fighting. The platelets are involved in clotting. All right, so RBCs. At least half of the volume of blood are red blood cells. And if you've ever had a value seen when you've had blood work done, one of the things that they measure, we actually do this in physiology here, is they measure something called a hematocrit, which is your percentage of red blood cells. And males, the average, the book says, the average percent red blood cells is about 46%. Females slightly lower, 42%. These values can vary a little bit. If this value goes down, well, either one of these values goes down, then you would be considered anemic, and perhaps, excuse me, a doctor would put you on, a diet that maybe might increase your red blood cell numbers. Red blood cells carry oxygen and a little bit of CO2. They actually surprisingly don't carry that much. You'll learn about that in physiology. They're shaped like little donuts. They have no nucleus. They're just chock full of hemoglobin. And I got another slide here that talks about that. But the average lifespan of RBCs in your body is about 120 days. So they're continuously being formed in the red bone marrow of your long bones. So the old ones are removed primarily by the spleen and by the liver. They're recycled. The hemoglobin is used over again or broken down and remade. So they don't live forever. 4 months, about right. Hematocrit's percentage of red blood cells are one of these measurements that are taken in various sporting events to make sure that somebody's not cheating and giving themselves more red blood cells than they might normally have. And in professional sports, if you're over a certain level in hematocrit, then you become under the magnifying glass of perhaps doing some illegal activities. In the old days, before erythropoietin was available, it was called